Hello everyone, today's lab is sickle cell Southern blot. The objective of this experiment is to determine whether the child of two sickle cell carriers have the trait or sickle cell disease. Their DNA was compared to three control DNA: person with sickle cell disease, person that is a carrier, and person with no sickle cell trait at all. Step 1 is to prepare 0.8% agarose gel. First you have the agarose, then the minor flask, then the distilled water and TEA buffer. You mix the agarose with the distilled water and the TEA buffer in the Erlenmeyer flask. Then you put the Erlenmeyer flask in the microwave oven to heat it for 30 seconds or so to make sure all the agarose is dissolved in the solvent. Once in the microwave, it heated, you take it out and you shake it to make sure all the agarose is dissolved. If not, you put it back into the microwave and heat for another 30 seconds. When all the agarose has been dissolved and the cloudiness has been gone, we put it on our table with a thermometer in it to let it cool to 65 degrees. Then we put it into our casting unit and we start pouring the agarose from one side, making sure no air bubbles are seen in the casting unit. We let the agarose to set and form a gel. As you see, the gels are formed over here. Step 2 is loading and running the gel. The line 1 is sickle cell gene sample, line 2 is sickle cell carrier sample, line 3 is normal gene sample, line 4 is mother's DNA, line 5 is child's DNA, and line 6 is father's DNA. After we have loaded all the samples into our agarose gel, we connect the electrodes to the voltage machine and set the voltage to 130 volts. We let it run for an hour. And we move to the step 3, which is Southern blot procedure. In this process, we depurinate the gel by placing it in a tray containing 200 milliliters of 0.25 molars hydrochloric acid. We leave it at room temperature for about 8 minutes. As you see, the gel is taken out into a little tray and 0.25 molar HCl is poured in. After 8 minutes has passed, the HCl is discarded away. And the gel is washed 2 times with 200 ml of distilled water. We make sure that the agarose gel is rinsed with several washes. Once the gel is washed, we then soak the gel in 200 milliliters of DNA denaturing solution, known as DDS, for 15 minutes on the shaker. After the 15 minutes is passed, we throw out the DDS. Then we take out the gel and place it on a sheet of plastic wrap on the tap. The gel is placed well side down directly on the plastic. Then the nylon membranes are left in the denaturation solution. Once they're soaked, the membranes are taken out and placed on top of the gel on the plastic. We make sure there are no air bubbles. Then we put a piece of filter paper on top of the membrane and we place a stack of paper towels. Along with a tray to put the beaker on top of it to apply pressure. Then we move on to step 4, which is the staining and detection. To detect the DNA, we flip the gel membrane filter onto the filter side. Then we remove the gel and allow the membrane to dry. We use forceps to avoid touching the center of the membrane. Then we place the dry membrane in a clean tray with 100 milliliters of dilute blue Blot solution for 10 to 15 minutes. So this is the blue Blot solution, which is used for staining. We put the gel tray on the shaker for about 15 minutes, and then we discard the blotting solution and rinse the gel with distilled water several times until the DNA bands are visible. As seen over here, the sheet is washed several times with distilled water. This is our final result. As we see, the bands are pretty visible. Both parents are carriers as indicated by the 2 bands. However, their child did not receive any of the sickle cell trait. Thank you for watching.